Okay, so the next thing we need to talk about are the actual muscle fibers and how they're going to work. So there's lots of new vocabulary that we have to talk about. So the first one is sarcolemma. Okay, so anytime sarco is in here, sarco refers to muscle. So the sarcolemma is just the plasma membrane around a muscle cell. Okay, the sarcoplasm is the cytoplasm in a muscle cell. Okay, now the endoplasmic reticulum in a muscle cell is called the sarcoplasmic reticulum. Its function is a little bit different. What it does is actually it's going to does similar things, but it also stores calcium. Okay, now if we talk about the structure of the muscle, okay, so I need to show you the next picture. So let me go through these, okay, and then I'll show you where they are in the picture. Okay, so if we start talking about terminology for a whole muscle, we can break it down from the big pieces down to the smallest piece. Okay, so the muscle fibers are arranged in bundles, and those bundles are called fascicles. So fascicles you could actually see with your naked eye. Okay, and then they're broken down into myofibrils. Okay, so myofibrils are bundles of myofilaments. Okay, myofilaments are individual groups of proteins, and the two proteins that we really talk about are actin and myosin. And actin and myosin are common in muscles of all sorts of animals. Okay, so myofilaments, they're in repeating units, and then the sarcomeres are like subdivisions of the myofilaments. Okay, so if we look at this, okay, so here's a muscle. Okay, so a muscle contains bundles of muscle fibers, and a muscle fiber has many myofilaments. Okay, so these little guys, this is a bundle of fibers. Okay, so each one of these would be a fascicle. Okay, so in these, okay, if we break them down, okay, so we take one of these little bundles, can we break it apart? Then we have a myofibril. Okay, so one myofibril is one of these bundles. Now if we take the myofibril and break it down, we have these strings which are called myofilaments. A sarcomere are these little units that repeat along the myofilament. I know, to me this is confusing too. Okay, so a because everything is, the names are all so similar. Okay, so the myofibril is repeating units of sarcomeres. Okay, so if you break them down by size, the sarcomere is the smallest that we're talking about right now. Okay, and a myofibril, then we're going to have a myofilament. Okay, and then we'll have the fascicle. Okay, now here's the sarcomere. So this is where all the good stuff occurs. So the sarcomere is where we're actually going to have muscle contraction. Okay, so when the muscle contracts, it gets shorter. So let me show you on here. So the sarcomere is made up of two protein myofilaments. Okay, so two strings of protein. One is called myosin. Okay, and myosin are these thick things in the middle. Okay, and myosin has these little things called myosin heads on them. So these little things that are sticking up, they're kind of like golf club heads, and those are called the myosin heads. Okay, actin is the thin filament. So these guys here, these are actin. Okay, the things that look like twisted up beaded necklaces, they're red. That's the actin. Okay, now when your muscle contracts, these filaments are going to slide over each other, and they're going to shorten this place called the H zone. Okay, so when everything's relaxed, when your muscles are relaxed, there is this zone in here that's called the H zone. Okay, and when muscles contract, okay, they slide past each other. Okay, the actin slides past the myosin. Okay, and it eliminates that H zone. Okay, and that means it's contracted. Okay, so the way that this happens, okay, it's called the sliding filament model. So the first thing that happens is a nervous impulse is going to travel down a motor neuron, and it's going to go to this place called the neuromuscular junction. So if I show you on here, okay, so here's a muscle, okay, here's the neuron coming in, okay, and coming off of the neuron are these little axon hillocks, okay, the axon terminals, these end pieces. So they're going to come up and they're almost going to touch the muscle. But so what's going to happen is the nerve is going to tell the muscle how to contract. Okay, so the nerve impulse travels down this neuron, okay, and then when it gets there, what's going to happen is it's going to release a neurotransmitter called acetylcholine. Okay, so, and we symbolize acetylcholine, ACH. Okay, so what it does is it passes through this space, okay, in between, okay, and it's going to bind to receptors on the muscle. So let me go back. Okay, so it's going to bind to receptors on the muscle, and when it binds, it causes these fibers to release calcium. Okay, so calcium is going to be important. Calcium is going to be released from the sarcoplasmic reticulum. So if I go here, okay, so here's the neuron, okay, and this neuron splits and it's going to go to a bunch of different muscle cells. Okay, so we're going to trigger all of them to be released at one time. Okay, to contract at one time. So here's the end of the neuron. Okay, and in it are little vesicles. Now this little green stuff in here, these are vesicles, so those are just like chambers in the neuron, okay, so in the nerve cell, and they're going to release acetylcholine. Okay, so acetylcholine gets exocytosed out, okay, it crosses the space in between the neuron and the muscle, which is called the synaptic cleft, crosses that, and it's going to bind to these channels. Okay, so acetylcholine comes over and it binds. Okay, now next thing that happens, okay, so we release calcium. When it binds, it's going to cause a neural impulse to travel down the muscle cell, and we're going to release calcium. So the calcium is going to come in, it's going to bind with this molecule, this protein called troponin. Okay, now troponin is wrapped around actin. I'll show you in a minute. Okay, and when that happens, it causes these tropomyosin threads that are covering actin to shift. Okay, and what's going to happen is the myosin heads are going to go into these sites. Okay, so the ATP will then bind to the myosin heads, and it's going to be used as energy to pull. That's what actually makes it come closer together. Okay, so let me show you. Okay, so here's the actin. So here's the red beads. Okay, they didn't show you this on the last picture, but what's actually around the red beads is the tropomyosin. Okay, so tropomyosin is the string, so I remember the S is for string. Okay, so tropomyosin's the string. Troponin, okay, are these balls. Now on the actin, we have little active sites, so there's little openings in there. They're normally covered up by this troponin and tropomyosin. If we release calcium, what's going to happen is it comes in, okay, so here's calcium, it binds to the troponin, okay, and it opens up these active sites, and they don't show you that on this picture, but it opens up holes here. So what's going to happen are the myosin heads, these little golf club things, want to go into those holes. So when the calcium comes in, it moves these out of the way, and the myosin heads hook into there. When they hook into there, what happens is it forms a cross bridge. Okay, ATP is going to be used to move it. Okay, so put the myosin head in there, we put it in, and it just kind of cocks it, almost like if you were pulling, if you're playing tug of war with a rope and you pull it, so your hands are the myosin heads, okay, holding on to the actin, and we pull it, and then you put your hands up and you move it down the rope and you pull again. So that's called a power stroke, and that's how we're going to slide it and how we're going to move the sarcomere pieces closer together. So it shortens that band right in there. I'll find a video and post for you guys. Okay, now, what role does ATP play in muscle contraction rigor mortis? Okay, so we need ATP in order to attach and detach the myosin heads. Okay, so if you don't have ATP, the heads are not going to come out of the troponin or out of the actin. Okay, so ATP is needed to attach and detach the myosin heads from the actin. So after we die, the muscle cells will continue to produce ATP and the muscles will continue to contract. Okay, but what happens is when we run out of ATP, some myosin heads are stuck in there, so there's no ATP for them to get loose, and that's called rigor mortis. Okay, so rigor mortis is when the body is stiff. There's still enough ATP in there to keep everything attached. So body temperature, the extent of rigor mortis is how, CSI, how you figure out, a forensic investigator will calculate the time of death. Okay, so